Hi there, Miss Vance again. This is the last part of Unit 9 Notes, Part 4, which goes into a little more detail about crossing over and some accidents that can happen during meiosis called nondisjunction. So when we talked about before, remember that in meiosis, the homologous chromosomes are going to get separated from each other in meiosis, and this can lead, of course, to differences between gametes, which we want. It's a big advantage to any species to have a lot of variation within the gene pool of an individual and of the population. Remember also that homologous chromosomes can have different versions of the gene at the same locus. You can have a brown-eyed gene from mom and a blue-eyed gene from dad, and that can get mixed up with other genes, and so you have a lot of different combinations. Since the homologous chromosomes also move to opposite poles during the anaphase, then the gametes are going to receive either the maternal or paternal version of the gene, and so if everything stayed like it was to begin with, then you'd see something like this. The offspring would inherit either the brown coat, black eyes from the mother, or the white coat, pink eyes from the father, if nothing else happened. We know, of course, that crossing over does happen, so that leads to more variation. This is called genetic recombination, and this is really important, again, to the variability of the genes within a species. Crossing over, of course, like we talked about before, is by definition the exchange of corresponding segments between separate chromatids, non-sister chromatids on homologous chromosomes. Okay, they join at a place called a chiasma. Chiasma is one of them. Multiple ones are chiasmata, and oftentimes they cross over more than one time. And then these corresponding amounts of genetic material are exchanged between the maternal and paternal chromatids. Now, how does the cell, or how do the chromosomes know where to separate? I can't tell you that because I don't really know. There are some thoughts about perhaps there are some signaling molecules that are attached at certain places in the genome to indicate this is a good place to split. I really wish I could tell you. As far as I know, it doesn't happen generally between or in the middle of a gene. It usually separates genes entirely. But here we have a diagram and also a micrograph of showing the chiasmata where the crossing over is occurring between those non-sister chromatids in the tetrad. Just a little more detail. Again, this is not something that you need to know really, but I thought it was interesting for you to be able to see it. That you can see even if you look at the at the level of the DNA strand, you can see the breaking that occurs there in specific spots. And again, there seems to be some specificity to where this occurs. I don't really know or understand how the chromosomes know where to divide, but they do generally divide in between genes. So here's what would happen with our two mice that we were looking at a while ago. So you have the maternal chromosomes that have the dominant traits for both coat color and eye color, and the paternal one, the blue one, that has the recessive alleles. Okay, and then when the breakage occurs in those homologous chromatids, the non-sister chromatids exchange portions at that chiasma. Okay, then you end up with chromosomes that have four different combinations of those different traits. So you have one chromatid that's going to have both dominant alleles, one that's going to be dominant for the coat color, recessive for the eye color, one that's recessive for the coat color and dominant for the eye color, and one that's recessive for both traits. And so what you'll end up with is two parental types of chromosomes that are just like the parents, but then you have two others that are recombinant, combining those traits in different arrangements, so that you have four different, very unique gametes that can be a result of this particular crossing over. Now, there are sometimes some things that can happen in the chromosome number and structure. There are some mistakes that can happen, and so we're going to talk a little bit about that and how that can affect the individual that results from that. First thing you need to know is when you look at a picture of the chromosomes, you can see some of these variations here. This is a picture called a karyotype. This is a picture of all of the chromosomes arranged in pairs. You usually produce karyotypes from dividing cells that get arrested at metaphase by adding a chemical that stops the formation of the spindle. And then you can take a picture, a photograph of the chromosomes, and then you can either manually cut the chromosome pictures apart, or you can, nowadays we do it digitally, of course, it's much easier that way. But this lets us look at the chromosome pairs and the number of chromosomes and the structure of the chromosomes, and we can sometimes see some differences there. Here's an example of a karyotype of a particular individual. Okay, notice that it shows the paired chromosomes, and these are, since they're arrested at metaphase, then we have the duplicated chromosomes, so it shows that kind of that X-shaped chromosome that we're used to seeing a picture of there. And you see that shows the centromere location and the chromatids and the homologous chromosomes like that. And again, you can use the banding pattern and the length and the position of the centromere to determine which one is which. We'll do an online Gizmos activity that lets you see kind of how some of this works next week when we're working through the rest of this unit. This individual has all 22 pairs of autosomes, and then the sex chromosomes, they have an X and a Y. And since there is only one X and then also a Y, that means this individual is a male. If there were 2 X chromosomes, it would be female. And if there was no X chromosome at all, there would not be an individual born, because you have to have at least one X chromosome in order to survive. Now, sometimes there are mistakes that happen, and you end up with an individual that has extra copies of chromosomes. There are a couple of ones, most of the time this happens. Actually the zygote or the embryo is not viable. It will die. It will be reabsorbed before birth occurs. But there are a couple of different times this can happen, that you have an individual that can survive. And so trisomy 21 is one of those examples. This is someone who's inherited 3 copies of chromosome number 21. It is definitely the most common human chromosomal abnormality. It causes Down syndrome. Most of us have had some experience with at least one person with Down syndrome. And so there are certain symptoms. It's a syndrome rather than a disease, because it's got a bunch of different things involved. But generally speaking, you have these characteristics that are included in a person that's diagnosed with Down syndrome. They have mental retardation to some extent. There are some characteristic facial features that they have. They're relatively short in stature. They don't get really tall. There often are heart defects and also susceptibility to certain kinds of infections or diseases like respiratory infections or Alzheimer's disease. And oftentimes they have a shortened lifespan. That is not always the case. I grew up with a girl who was Down syndrome, and she lived until her 60s. She was about 65 or 66 when she passed away a couple of years ago. And so it doesn't necessarily mean that you have all of these things. Some individuals are affected more than others. There's a lot of different things that can affect that. Okay. Of course, the incidence, or how often this occurs, it increases with the age of the mother, because oftentimes what happens is there's a mistake that occurs in the production of the egg cells, and that can lead to Down syndrome. Here is an individual with Down syndrome, and the karyotype for this individual. Okay, and you see that there, when you have the karyotype showing the ordered pairs of the chromosomes, there are 2 X chromosomes showing that she's female, and 3 copies of chromosome number 21, which is the diagnostic feature of trisomy 21 or Down syndrome. There are several others, and we're going to do an activity where you get to identify some of these things next week. A short little video about how non-disjunction can happen.
Do you know anyone with Down syndrome or Klinefelter syndrome? These are examples of chromosomal disorders, which are due to non-disjunction. When non-disjunction occurs during meiosis 1, replicated homologous chromosomes do not separate correctly. One daughter cell retains both replicated chromosomes. The other daughter cell lacks that chromosome. When a haploid sperm fertilizes an egg with an extra chromosome, trisomy occurs. When a sperm cell fertilizes an egg that lacks a chromosome, monosomy results. In this scenario, either a trisomy or a monosomy results. Nondisjunction can also occur during meiosis II. In this scenario, meiosis I occurs normally. When the daughter cells separate during meiosis II, one daughter cell divides normally, but the other daughter cell divides abnormally. One egg lacks a chromosome while the other egg has an extra chromosome. When a haploid sperm cell fertilizes a haploid egg, a normal diploid results. If the sperm cell fertilizes the egg that lacks a chromosome, then monosomy results. If the sperm cell fertilizes the egg with an extra chromosome, then trisomy results. In this scenario, there is a 50% chance of a normal diploid, a 25% chance of a monosomy, and a 25% chance of a trisomy.
So there are a couple of different ways that can happen, like we said, okay? By definition, nondisjunction is the failure of chromosomes to separate normally during meiosis. Chromosomes are chromatids. And it can happen either in meiosis I, like the video showed, or in meiosis II. Anytime you have fertilization after nondisjunction, then you're going to end up with zygotes that have different numbers of chromosomes, okay? So again, like it showed in the video, meiosis I, if nondisjunction occurs in meiosis I, then you have one of the tetrads that does not separate. And so you end up with gametes that have either a monosomy, will either produce a monosomy or a trisomy in this case, okay? A monosomy means having only one copy of the chromosome. In humans, there is only one survivable monosomy, and we'll talk about that. If it happens in meiosis II, then again, the homologous pairs are going to separate normally in meiosis I, and then if nondisjunction occurs in meiosis II, then one of those duplicated chromosomes gets pulled to the daughter cells, and you end up with one that's going to have a trisomy, one that's going to have a monosomy, and then the other two would be normal gametes that would be produced there. These are always going to cause problems. Anytime you have extra or fewer chromosomes, it's going to cause some kind of a problem, okay? There are also other alterations of chromosome structure that can cause birth defects and cancer of various kinds, and these are going to be a certain type. These are chromosomal mutations rather than gene mutations like we talked about when we talked about DNA, okay? These can produce genetic disorders or sometimes cancer in some kinds of instances, and so these would be things like a deletion of a part of a chromosome or a duplication of part of a chromosome, an inversion, which is when part of it breaks off and reattaches backwards, or a translocation, which is attaching a part of one chromosome to a separate, different, non-homologous chromosome. And sometimes it can be reciprocal, and you end up with two chromosomes that are trading parts that are not homologous chromosomes. And so this is just a picture. I think we talked about this before. We talked about gene mutations that we talked about when we talked about DNA. Gene mutations may be silent. They may not cause any problems, but chromosomal mutations like this are always going to cause some kind of a major problem. Generally speaking, either death or some kind of severe condition. Some of the trisomy type conditions can be produced by a duplication, where an extra piece of a chromosome is attached there, and that they're always going to cause a problem. There's one particular kind of cancer that can be produced by a reciprocal translocation between two specific chromosomes that we may have time to talk about in class, and we may not. This ends the notes for Unit 9, Part 4.